Welcome to this complete HISAT2 alignment and indexing tutorial for RNA-Seq data analysis. In this video, I will walk you through the full step-by-step workflow, from building the genome index to aligning paired end reads and analyzing the results. This tutorial is beginner-friendly, practical, and designed for both Mac and Linux users. Let's get started. Alignment and indexing. In RNA sequence analysis, two important steps are alignment and indexing. Alignment means mapping sequencing reads from FastQC files back to a reference genome. We use tools like HISAT2 to find where each read belongs in the genome. The output of alignment is usually a SAM or BAM file, which tells us the exact genomic coordinates of each read. But before alignment, we must build an index. Why? Because genomes are very large. Searching directly inside a huge genome file would be slow. Indexing creates a fast lookup system, like a table of contents in a book. When we run HISAT2 build on a genome faster file, it creates index files such as 1.ht2, 2.ht2, and so on. So first we build the index, then we align the reads. In this slide, I am showing the structure of a basic bash script used for RNA-Seq alignment using HISAT2. First, we start with the script header. #usr bin env bash. This line tells the operating system to use the correct bash interpreter installed on your system, whether you are using Mac or Linux. Next, we use a safety command. Set your pipe fail. This makes the script safer and more reliable. If any command fails, the script will stop immediately. It also prevents errors from undefined variables and pipeline failures. Now, the script is designed for HISAT2 RNA-Seq alignment using Conda and Samtools. We define the environment name. env and ame equals HISAT2_env. This is the Conda environment where HISAT2 and Samtools will be installed. Next, we define the input FastQC files. These are paired end sequencing reads. R1 equals SRRR1, FastQ, GZ. R2 equals SRRR2, FastQ, GZ. Then we specify the reference genome file. genomefa equals genome, FA. This is the faster file used to build the HISAT2 index. After that, we define output and index naming. IDXPREFIX equals genome index. OUTPREFIX equals sample HISAT2 inches. These names help organize alignment results. Finally, we specify the number of threads. Threads equals 8. This allows parallel processing, making alignment faster. So this section of the script mainly defines inputs, outputs, environment, and execution settings before running the actual HISAT2 alignment. In the next step, we will use these variables to build the genome index, run alignment, and generate sorted BAM files. In this slide, I am showing the complete step-by-step workflow for RNA-Seq alignment using HISAT2 and Samtools. Step 1. Install and activate environment. First, we create a Conda environment that contains HISAT2 and Samtools. We run Conda create env and amec Conda for HC Bioconda HISAT2 Samtools y. Then we activate the environment. Conda activate env and ame. This ensures all required tools are installed and isolated for this project. Step 2. Build HISAT2 index. Next, we build the genome index using the reference faster file. HISAT2 build genome, FA genome index. This step converts the genome sequence into an indexed format that HISAT2 can use for fast alignment. Step 3. Align RNA-Seq reads. Now we align paired end FastQ files to the reference genome. HISAT2p threads x genome index -1 R1, FastQ, GZ-2 R2, FastQ, GZ as sample.sam. Here, threads control speed. Genome index is the index prefix. R1 and R2 are paired end reads. Output is a SAM file. Step 4. Convert SAM to sorted BAM. SAM files are large, so we convert them into compressed BAM format. First, Samtools view at threads bs sample.sam. Then we sort. Samtools sort at threads o sample, sorted.bam. Finally, we index the BAM file. Samtools index sample, sorted.bam. Now the alignment file is ready for downstream analysis and visualization in IGV. Step 5. Alignment QC statistics. To evaluate alignment quality, we run Samtools flagstat sample, sorted.bam. This generates statistics like total reads, mapped reads, properly paired reads. These metrics help confirm whether alignment worked correctly. So this workflow covers the complete RNA-Seq alignment pipeline. Environment setup index building read alignment BAM processing QC. In the next step, We will use this BAM file for downstream analysis like differential expression or visualization. Now we are inside the terminal, and this is a working directory where the RNA sequence FastQ files and other analysis folders are present. Next, we are going to create a Conda environment specifically for HISAT2 alignment. Using a separate environment is important because it keeps all bioinformatics tools organized and avoids software conflicts. Now I am running this command. Conda create nHISAT2 _envc Conda FastQC bioconda HISAT2 Samtools y. This command will create a new environment called HISAT2 _env. Install HISAT2. Install Samtools. And pull packages from Conda Forge and Bioconda channels. Now the installation is complete, and you can see all the packages have been downloaded and installed successfully. Now, I am deactivating the current environment to demonstrate how environment switching works. Next step, we will activate the HISAT2 environment and confirm that HISAT2 is installed correctly before starting the alignment process. First, I am checking the available files using the ls command. Before alignment, we need to prepare the reference genome for indexing. So I am creating a new folder called index_files to keep all HISAT2 index files organized. Next, I am copying the genome FASTA file from the NCBI dataset folder into this index directory. This genome file will be used to build the HISAT2 index in the next step. Next, I am also copying the genome annotation file, the GTF file, into the same folder. This GTF file contains gene annotation information, which will be useful later for transcript assembly and gene expression analysis. You can see both important files are present. genomic.fna, reference genome. genomic.gtf, annotation file. I am confirming the HISAT2 installation by checking its version. This confirms HISAT2 is installed and ready to use. I am running the HISAT2 build command using the genome FASTA file as input and giving a prefix name for the index files. This process will generate multiple index files with extensions like ht2. This step maps our RNA-Seq reads to the reference genome, allowing us to determine where each read aligns. This process takes lot times. Once the alignment finishes, we will convert the SAM file into a sorted BAM file for downstream analysis. These are the HISAT2 index files. You only need to build this index once for a given genome. Here I am installing SRA tools and pigs, which will help us download and process FastQC sequencing data efficient. After installation, I'll verify whether required tools are available. Here, when I run datasets, version. Next, I check the SRA toolkit installation using prefetch, version. You can see the version is displayed, which confirms that the tool is correctly installed. Now we are on the NCBI SRA webpage. Here you can see the RNA-Seq dataset information. This dataset is from EADS Egypti, and it contains sequencing data generated using paired-end RNA sequencing. This accession number, starting with SRR, is what we will use in the terminal to download the raw sequencing data. In this example, I will copy the SRR accession ID. Now we will go back to the terminal and use the prefetch command to download this dataset directly from NCBI. This command connects to the NCBI server and downloads the raw sequencing data. You can see it is resolving the accession, checking the format, and downloading via HTTPS. Here it shows that the download was successful. It also confirms the file size in bytes and that the accession is valid. Now the raw SRA file has been downloaded successfully into our working directory. You can see here that the accession SRR3721189 was resolved, validated, and downloaded from the NCBI service using HTTPS. This means the raw sequencing data is now stored locally in SRA format. The next step is very important. We now convert this SRA file into FASTQ format using the FastQC dump command. Next, I use the command FastQC dump SRR3721189, split files, threads 8 here. Split file separates paired end reads into two files, R1 and R2. And threads 8 speeds up the conversion using multiple CPU cores. To save disk space and make them easier to manage, I am compressing both paired and FASTQ files using the pigs command. After this step, the files will be converted into FastQC GZ format, which is the standard input format used for HISAT2 alignment and most RNA-Seq pipelines. Before performing alignment, we always check the read quality using FastQC. So here, I am installing FastQC inside the same HISAT2 environment using Conda. This will install FastQC along with all required dependencies from Conda. Now FastQC is installed, and we are running quality control on paired end file one by one. You can see the analysis has started, and FastQC is showing the progress percentage as it processes the file. Now FastQC analysis is completed. I am opening the HTML report file to visualize the quality results in the browser. Using the command open srr372118391_1_FastQC, HTML. Now the FastQC report has opened in the browser. This summary panel gives a quick overview of sequencing quality across multiple metrics. On the left side, you can see modules like per base sequence quality, GC content, duplication levels, and adapter content. Green check marks indicate good quality, while warnings or failures highlight areas that may need trimming or further attention. Now that quality control is complete, we are ready to perform alignment using HISAT2. Here, I am running the alignment command. Let me briefly explain this command. P8 specifies that we are using 8 CPU threads to speed up the alignment process. Index_files, EADS_index is the reference genome index we built earlier. -1 and -2 represent the paired and FASTQ files, forward and reverse reads. And S defines the output file, which will be generated in SAM format. This step maps our RNA-Seq reads to the reference genome, allowing us to determine where each read aligns. Once the alignment finishes, we will convert the SAM file into a sorted BAM file for downstream analysis. Great! The alignment step has now completed successfully. You can see the total number of reads processed, and how many aligned concordantly, meaning both paired reads mapped correctly to the genome. Some reads aligned exactly one time, some aligned multiple times, and a small fraction did not align. At the bottom, we can see the most important metric, the overall alignment rate, which is about 94%. Now that alignment is complete and we have generated the SAM file, the next step is to convert it into a compressed and sorted BAM file. Here, I am running the command. Samtools view BS converts the SAM file into BAM format, which is a compressed binary version and much smaller in size. The @8 option tells Samtools to use 8 threads for faster processing. Then we pipe the output directly into Samtools sort, which sorts the alignments by genomic coordinates. The O option specifies the output file name for the final sorted BAM file. As you can see, the SAM file has now been successfully converted into a sorted BAM file. In the next step, we will create an index for this BAM file to allow fast access to specific genomic regions. I just want to clarify one small thing here. In the previous Samtools sort command, I did not explicitly include the word sorted in the output file name. But technically, this file is already sorted because we used Samtools sort. That's why I am now renaming it to SRR372113839_HISAT2 sorted.bam. Now that our BAM file is sorted, the next step is to create an index file using Samtools. You can now see that the sorted BAM file and its index file have been successfully generated. Before moving to downstream analysis, it is always a good practice to check alignment statistics from the final sorted BAM file. Here I am running Samtools flagstat command. The flagstat command provides a summary of alignment statistics, including total reads, mapped reads, properly paired reads, duplicate reads, secondary alignments. The @8 option uses 8 threads for faster processing. Now I am checking the alignment quality statistics using the Samtools flagstat output file. Now I have opened the alignment summary file generated using Samtools flagstat. At the top, you can see the total number of reads processed, including both QC passed and QC failed reads. The most important line here is the mapped reads percentage, around 94% of reads mapped to the genome, which indicates a strong and reliable alignment. In this video, we covered the complete HISAT RNA sequence workflow step by step. We started by setting up the Conda environment and building the HISAT2 genome index, then aligned paired end reads to generate the SAM file. After that, we converted SAM to BAM using Samtools, sorted and indexed the BAM file, checked alignment statistics with flagstat. Thank you for liking, commenting, and subscribing. I truly appreciate your support.